pleasure to start the meeting, and I would like to introduce Karl. I think Karl, everyone knows. He's a homebrew Londoner. He worked a long time at UCL, doing amazing work in BMT, but also in cellular therapy, CMV. I think you've been everywhere, Karl, based on what I've seen in your work. But he will give us a new insight on his new topic. Karl, please.
Thank you very much. And thank you to the organisers for the kind invitation. It's absolutely fantastic to see some faces I haven't seen for a number of years. And I've been indulged today. I was just saying, it's a long time since I had more than 12 minutes to speak. So this is something of a novelty for me this morning. And what I've come to talk to you about at the basis is tumour neoantigens, but I'll go a bit beyond that and touch on a number of the themes that were touched on yesterday. Achilles Therapeutics, where I am the Chief Medical Officer as of last year, and Roche, and that will become relevant for some talk about some development of an anti-CD25 antibody. I just thought I'd indulge myself a bit further and give those of you who don't know me a sort of start of 10 about my background and how I got to where I am now. In 1991, I qualified from Oxford University. At the same year, my brother developed acute promyelocytic leukemia. That's him up there with my sister. And he was one of the first people in the UK to get all-trans retinoic acid. It was shipped from London, from the Hammersmith, in fact, down to Oxford, where he was being treated. Of course, my sister and I were dutifully tissue typed because standard of care at the day would have been an allograft in first remission for APML. Thankfully, we didn't match, actually, because we all know what happened subsequently with the sort of revolution in treatment for APML with all-trans retinoic acid. And that really is a theme, then, that I've sort of carried through much of my career, this idea that, you know, there are transformative moments where we can really touch and change the lives of patients. I was very fortunate in 2004 to go to New York, where I met a number of other new and good friends now, to work with James Allison, who, of course, was working on CTLA-4 blockade. I was put in touch with Jim by Paul Moss, who's in the audience today, and hugely thankful to him always for that first introduction and telling me that CTLA-4 was an interesting thing for me to look at. So I spent 3 happy years in New York with Jim. He, of course, subsequently got the Nobel Prize for his work on CTLA-4. At that time, I met Sergio Casado, who was a postdoc in Jim's lab, and we worked very closely together. Following my return to UCL, I did manage to convince him, after about 3 years of trying, to come over, and we set up a joint lab in 2010 at UCL, where we co-researched for over a decade, and I'll share some of that research with you. In 2010, we met Charlie Swanton, and I'll touch on his work. He's well known to many of you as Mr. Tumour heterogeneity, and it was through that and the work on clonal neoantigens in particular that we sort of came together with the interface of his clonal evolution and our views on immune response and prosecution of neoantigens with therapies. So a number of these themes came up yesterday. There are many factors, obviously, that influence both the efficacy and potentially the toxicity of cellular therapies. These, I think, all came up in a variety of different ways yesterday: microenvironment and trafficking of T cells or alternate effective cells to the tumour. Cell function, that is cell fitness or enhanced cell function. We've all seen the importance of enhancing cell function with CAR-T, with that co-stimulatory domain. And then there was a lot of talk about target quality. And I think without quality targets, to some degree, a lot of the rest of this doesn't matter. If you haven't got a good target, it doesn't really matter how fit your cell is. It doesn't matter how much it can ingress into the tumour if your target quality is low. And when we think about target qualities at a very high level, we can make a number of relatively facile statements. An ideal target should be only present on malignant tissue. That obviously will allow us to specifically target the cancer and avoid the collateral toxicities. Failing that, we go down the CD19 route, it must be present on an expendable tissue compartment. Now, that works for B-cell malignancies, but as we heard yesterday, not so much so for solid tumour malignancies, and that really is the Everest here. How do we get cellular therapies to work in patients with solid tumour malignancies? An ideal target should have a high and homogeneous expression level. Easier said than done again. CD19, perhaps a good example of fulfilling this criteria in the main. But as we heard yesterday, a number of the solid tumor antigens are relatively heterogeneously expressed, particularly those cancer-associated testis antigens, the tumor-associated rather than tumor-specific antigens, which both are present heterogeneously and are present on normal tissues, so they don't fulfill the first criteria. Now, that doesn't mean they're not valuable targets. It doesn't mean that there aren't ways to work around that, but they're probably not the ideal target. And then finally, at a high level, that target should not be lost through tumor evolution. Again, well exemplified by CD19 in B-cell malignancies, but also true potentially of cancer neoantigens. Loss of heterozygosity of the MHC is well described in a variety of malignancies, and that at a single stroke can rid you of a number of your potential cancer neoantigen targets. If we're failing that sort of criteria for loss of single antigen through tumor evolution, then ideally our therapeutic should encompass multiple targets. So if we have a uni-targeted therapy and we lose that target, obviously we lose therapeutic efficacy. Whereas if we have a poly-targeting therapy, then maybe we can get around that. So thinking about this is the only picture I have of Sergio, Jim, and myself taken at 2 a.m. In the Nobel after party. But when we were working with Jim, we were working obviously primarily on function and mechanism of CTLA-4 blockade, but we began to think a lot about target antigens at the time. And we wrote a piece in Cancer Cell in the early days of the emergence of sequencing of tumor. So it was around that time that we saw the first sequencing of the first breast and colon cancer tumors with papers coming out showing the level of mutational diversity in those particular tumors. And we were lucky enough to get access to that data and did some, what now looks like very clunky bioinformatic approach to try and predict what neoantigens would arise from those mutations. And our thesis was essentially this. Nowadays, this is all taken for granted, but it really wasn't in those days, that it was the malignant genome and these mutations that were driving both class 1 and class 2 antigen presentation on the T-cell surface. And it was T-cells targeting these that we would eventually be able to hopefully enhance function by inhibiting those inhibitory checkpoints with drugs such as ipilimumab. And at the time, a drug called tislelizumab, which then became tremelimumab, and others. And at the time, 2006-07, PD-1 really was something of a distant dream. People really hadn't attached themselves to what became in the next decade, potentially the greatest immuno-oncology breakthrough that we've seen in solid tumors. We were also very interested, we put this in the corner, and there was a little piece, a paragraph in there about CD25 and ONTAC, which was a diphtheria toxin conjugated IL-2 binder, which essentially potentially would deplete Tregs, and I'll come back to that later as another mechanism of enhancing immunity. Now, we had these theories, and we wrote in that manuscript that we can make a number of predictions, but the arguments remained largely conjectural, and that's because we didn't have the technologies to really interrogate those questions. We didn't have high throughput streptomer screening to really look for immune response to neoantigens. We didn't have rapid and deep sequencing of vast numbers of tumors to actually get the basic bioinformatics. But what we were saying at the time was that we felt that the genetic instability that actually would prevent small molecule inhibitors from ever really making huge transformative change for the vast majority of patients with solid organ cancers were actually, at the same time, potentially a unique opportunity to provide targets for anti-tumor immunity via the generation of these neoantigens. Now, Charlie Swanton was coming at this from a slightly different angle. He was very interested in tumor heterogeneity, that is the difference in mutational signals you see in different parts of the tumor, which led to the theories or confirmation of some earlier theories of cancer evolution, whereby cancer is accruing mutations for a period of time before it has that sort of transformation event. And when it finally gets that transformational event and becomes neoplastic, those mutations are locked in up until that time point. So those are what we call the clonal or trunk mutations. Of course, subsequently, the tumor can mutate further, and subclones will be derived, which have new mutations, but they will carry these red mutations, these clonal or trunk mutations, through to the daughter cells. So the argument from Charlie was, firstly, that despite all these mutations, very few of them will actually be potentially targetable by small molecule inhibitors. And if they happen to be in the branches, you're never going to cure the cancer if it can just grow up another branch off the trunk. So he was a bit of a loss, he'll admit, thinking that he'd done all this beautiful work, but essentially it was predicting that the world of small molecule inhibitors wasn't going to really deliver the vast swathe of cures that was hoped for at the time when small molecule inhibitors were emergent. And we came together because you can obviously see some similarities here. Of course, the world of genomics have moved on. Charlie was doing a lot of sequencing of a variety of tumors at this stage. And we put our immunology hat on and said, okay, we've made these predictions earlier on. If what we said is true, then there should be an association between mutational load, tumor mutational burden, and response to checkpoints. And he and others in other groups demonstrated that was indeed the case. If you look at patients getting durable clinical benefit to checkpoint, they on average have a higher mutational burden overall than those who don't get a durable response. Now, focusing on Charlie's particular interest, a couple of outliers here who do have a high tumor mutational burden but don't get durable response, it was just very interesting to note that a large fraction of their mutational burden was actually subclonal, the branches. So targets that you wouldn't predict actually would beget long-term durable response. We were able then, through the development of the high-throughput streptomer screening, to load multiple of these hundreds of potential neoantigens onto HLA molecules and do high-throughput screening of the samples that Charlie was generating from the TracerX. These are early-stage lung cancer patients resected with curative intent. He was doing his bioinformatics, but we got access to the tissue to look for immune response. There was also always paired normal tissue from next to the tumor. And what we were able to demonstrate that we could see T cells responding to tumor neoantigens within the tumor that were actually specifically enriched in the tumor and not seen in surrounding normal tissue. Now, take a step back and think about TIL therapy, because this is a cellular therapy meeting, not a checkpoint meeting. We're all aware that TIL have been around for an awful long time, since the 80s. The basic idea here is you excise tumor, you chop it up into a number of smaller lumps, you then expand the T cells out of that tumor, do a much further sort of harder expansion with high doses of IL-2 and polyclonal stimulants, and generate a T cell product that you can then reinfuse back into the patient. Now, again, if our predictions are right, the active component of this should largely be neoantigen reactive T cells. And here's an example of a lesion in the upper thigh of a lady that was resected to make the TIL product, and then what is seen in a minority of patients, a very dramatic clinical response with resolution of the tumor lesions. Now, this really, in the early days, was only demonstrated to work specifically in melanoma. Melanoma, of course, has one of the highest tumor mutational burdens of all cancers, so you would think is a relatively low-hanging fruit for a neoantigen targeting therapy. Now, all of this work, of course, was performed by a number of people, but pioneered by Steve Rosenberg. And in these earlier days, these were the sort of curves that we saw, that really, as we see with CAR-T, a sizable minority of patients are getting durable clinical benefit. In fact, in the earlier studies, overall response rate was in excess of 50%, and 25 to 30% were getting durable clinical benefit. And the message at the time was that this response was perhaps agnostic to prior therapy, so it didn't matter what prior therapy you'd had, be that high-dose IL-2 or indeed anti-CTLA-4. The other striking thing here was about 20% of those early patients were attaining CR, and those patients who attained CR really did remarkably well. Now, it's taken a long time to get TIL almost to a licensed product, and the one that's almost there is this one, Lifileucel, that's come through a company that's evolved over time. In 2007, Genesis was formed as a company to prosecute TIL in melanoma in particular. In 2013, it fused with another company and became Lion Therapeutics, and there's their logo. And then in 2017, it evolved further to Iovance, and look, they even kept the same font. But way on back in the early teens, they had this study design, essentially with cohort 1, a fresh TIL product, cohort 2, a cryopreserved, and cohort 3, a retreatment. It's then taken another almost decade to get to the point where Iovance have taken that study on, increased the number a bit. This is cohort 2 now, presented in JCO last year, where they've reached, sorry, Andrew, 78 patients enrolled, 67 treated. This is the treated patients. You can do the maths yourself. I couldn't redraw the curve last night. So 67 received TIL, and if you look at the response rate, around about a third of the patients get a response. So not as good as the early days that we saw where it was said to be 50%, and there may be a number of reasons for that. And here's the progression-free survival. And in the responders, a far smaller number now are CRs. It's less than 10%, so the majority of responders are PRs, but in the responders, a significant duration of response. Now, they've recently updated with a cohort 4, the FDA-mandated sort of licensing cohort, which essentially is the same as cohort 2, but now delivered between 2019 and 2020. So less historical patients, and treatments have changed. The results aren't quite as good, and there are potentially a number of reasons for that, which we'll look at. So as I said, that you can make a prediction that TIL response should look like CPI response. There should be a relationship between mutational load and durable clinical benefit. Patient numbers are smaller here. This kind of work took quite a long time to evolve and come out, but the message is there. If you look at the patients getting clinical benefit, on average, they have higher mutational burden, and those with high mutational burden survive better. Now, of course, if you think about the immune response system, it's not quite like a small molecule inhibitor where you're trying to target a cancer driver, if you will. The immune system is kind of agnostic to whether your mutation is a bystander or in an important mutation, an important gene that drives that oncogenic process. So it's interesting to see then that driver gene mutations and copy number aberrations actually were not associated with clinical benefit. The immune system doesn't care where the mutation is. It can be anywhere, which gives you this much broader array of potential targets if you're thinking about therapeutic. Now, there are a couple of very well annotated examples that demonstrate the potential power of neoantigen-reactive T cells in other solid organ malignancies. This from Eric Tran, a very famous paper for those in the field. So essentially, a patient with metastatic cholangiocarcinoma had their lesion resected, had a TIL product made. Interestingly, the way you make TIL product, you make multiple bags, and this product was made up of sort of 4 bags pulled into one. Retrospectively, they were able to identify the actual mutations and then the T cell responses, and essentially they had a T cell response against a single mutation. And in the bag that they had infused, about a quarter of the total product was actually neoantigen reactive. And they got something of a clinical response, a demonstration of activity. 6 months later, the patient progressed. By this time, they'd done all their retrospective work, and they fished out the cells from the bag. There was one bag that had about 95% clonal reactivity and 3 bags that had very little, and that's one of the difficulties of classic TIL expansion. It's very stochastic. You don't get to direct antigen-specific expansion. You get what you get. Sometimes you get a good bag, sometimes you get a bad bag. But they took this good bag, if you will, re-expanded the cells, so they had a bag of 95% tumor-reactive cells, and re-infused those again with a second response. So evidence that in the right patient with the right target antigen, you can see clinical activity directed seemingly by neoantigen response. And that's just another example in another disease, a lady with breast cancer, where, long story short, in this case, there were 2 neoantigens targeted by the T cells, again making up about a quarter of the bag that was infused, with regression of all of her metastatic breast cancer lesions, and really quite a durable response. By now, the field had moved on. This work is from Sidney Hadrut, working with the group in Hairlive, who are very active in the TIL field. From our earlier predictions, again, you're going to make a number of predictions leading into the world of TIL. And what these guys were able to do is they were able to look at A, the number of neoantigen responses, and B, the size of the circle, the frequency of response. So the higher you are up the y-axis, the more neoantigens you're targeting, the bigger the circle, the more the total product is made up of neoantigen-reactive T cells. And if they then take that data and analyze according to whether the patient's got a response or not, it's quite striking. These are only CD8 responses. So there's an additional element here of CD4 that we're not capturing. But patients who were getting primary progressive disease very rarely had either a significant number of reactivities or a significant fraction. And indeed, if you look at response according to number of NARTs above and below median, or frequency of NARTs above and below median, and this is the tertiles, you'll see that higher numbers of NARTs, so higher number of targeted neoantigens, or higher fraction of product targeting those, are associated with better outcome. Particularly striking, numbers are very small here, but with the number, the frequency of cells within the product. So on the basis of these, you can make a number of other predictions or hypotheses that optimizing both an RT number, that is the number of reactivities, the number of clonal neoantigens you're targeting, and the frequency may improve clinical responses to TIL. There's another issue that we talked about a bit yesterday, and that's cell fitness and cell function. And when we think about cell fitness, again, a rather simplistic cartoon, but as T cells differentiate, they lose proliferative capacity. At the same time, they gain effector function until you push them too far and they sort of fall off the cliff, become terminally differentiated and lose effector function. And the early TIL kind of sat here. They'd been hugely expanded to get very high cell numbers. They had probably relatively little proliferative capacity, and they were dropping off the cliff. The adolescent TIL that came later, that we kind of see in the more modern studies, were cultured for a shorter period, not pushed quite so hard, and were pushed back to regain certainly some of the effector function, although probably still losing quite a degree of proliferative function. But we knew from the world of antiviral T-cell therapies that actually if you used antigen-specific expansion, you could shift yourself down to here, where you potentially were maintaining both proliferative capacity and a decent amount of effector function. So if you're going to think about a next-generation TIL, then knowledge of antigens will allow you potentially to get yourself a fitter product to infuse less cells or do more once they get into the patient. Another issue I wanted to touch on is IL-2 addiction. So the sort of heroic expansion of the early generation of TILs was enabled essentially by the use of high doses of IL-2. The IL-2 receptor is a trimeric receptor, the alpha chain being CD25, the high affinity receptor. IL-2 can also, the function of the IL-2 alpha receptor can also be provided in trans by antigen-presenting cells, dendritic cells and the like. Now, when you think about what was happening in the TIL co-cultures with very high doses of IL-2, what you were getting over time essentially was basically signaling through the low affinity receptor, downregulation of CD25, the high affinity receptor, and a cell that proliferated was happily signaling internally, but was now addicted to high doses of IL-2. So when you put it back in the patient, if you took away the high dose IL-2, you lost the signaling and the cells died, which meant you had to give the patients high dose IL-2, which, as any of you who are active in the field know, is quite toxic. And certainly if you're moving away from younger melanoma patients to older lung cancer patients, that's a problem. So another thing you would like to do to enhance function if you're generating a next generation therapy is to avoid this IL-2 addiction, avoid the need for high dose IL-2 following infusion, avoid therefore the IL-2 associated toxicities, hence access more of your patient population and potentially facilitate ambulatory delivery. And that's really where we've gone with Achilles. This is the only specific Achilles side conflicts, as you know. But essentially the idea is, as outlined earlier, you start with these clonal neoantigens that are then shared down through the life of the tumor, such that these red antigens are present potentially on all tumor cells. They're neoantigens, they're not present on normal cells, so they fulfill that criteria of an ideal tumor target. And what we're doing is essentially doing the genomic piece, predicting the neoantigens, making the peptides. You could do that with tandem mini-genes or with free peptides, feeding them to a professional antigen-presenting cell, and then co-culturing them with the T cells we extract from the tumor. And what that does is it directs expansion of the antigen-specific, clonal neoantigen-specific T cells that we can then refuse to patients. That's not the only way to prosecute neoantigen reactive responses. This is a very recent paper in New England Journal. Switch stuff now, where essentially they've transduced T cells with two T cell receptors that target the mutation in KRAS. So pancreatic cancer, as we all know, has an appalling outcome when it's metastatic, but relatively high frequency of KRAS mutations. And in this elderly lady, what they were able to demonstrate was HLA C0802 restricted, or she expressed 802, and they were able to transduce with AO2 restricted TCRs targeting either a 9-mer or a 10-mer. And following some slightly unusual preconditioning, they infused cells followed by high-dose IL-2 in this case, and were able to demonstrate regression of visceral metastases with an overall PR lasting at least 6 months. Interestingly also, the engineered T cells continue to represent a relatively large fraction of circulating peripheral blood at 6 months. Now, there was a second patient in the paper also treated who didn't get a response, but an early demonstration that TCR redirected therapies potentially can target, if you've got the right target, cancer neoantigens and potentially beget important clinical response. Now, the problem here is that public or shared neoantigens are relatively rare. So most mutations that we see as neoantigens are private mutations, and so this really is a patient-specific approach, and it's relatively rare that you'll find a mutation that is expressed frequently enough amongst different patients in the context of a shared HLA that allows you to access a large number of the patient population. So this is expressed by about 8% of white Caucasians. So even in the best case scenario, you're less than 10% of your patients you can potentially treat, and they have to have the appropriate mutation. Now, the world moved on somewhat in terms of therapies for underlying disorders, and what became quite apparent from a number of groups was this rather striking factor, that if you looked at TIL therapy and the impact of prior PD-1 blockade, those patients who were PD-1 refractory were doing a lot worse than those patients who had never seen PD-1 before or weren't PD-1 refractory. This is also true of patients who are BRAF or MEK inhibitor refractory. Although the mutation begets relatively little differentiation of the curves, if you receive those agents and you become refractory, your chance of responding to TIL is very low. Now, this is a large NIH pooled data cohort. They were able to recapitulate the early data showing that prior anti-CTLA-4 didn't seem to have this impact. But in the PD-1 refractory patients, there was a lower overall response rate and lower duration of response. And this probably partially explains why cohort 4 of that Iovance study doesn't look as good as cohort 2, because times have moved on. So let's just do a little thought experiment for a moment about responses to checkpoints and responses to TIL. We can think about patients in a number of ways. There could be patients that have essentially no neoantigen-reactive T cells ever in their tumor. They didn't have the right mutation. They didn't beget an immune response. There are patients who have NAR-T in their tumor, and you can beget a response with a checkpoint inhibitor. And then there's another group of patients here who do have NAR-T, but they're non-responders. And the question is, can we improve these responses with other checkpoints and other modalities? These patients are potentially never responders to a TIL-based therapy. There may be other ways to beget response. Of the patients who respond to checkpoint, of course, some of them are durable responders, and they'll stay on checkpoint. So they're not going to fall into your cohort that you then treat with your TIL. So it's no great surprise that actually your PD-1 refractory cohort that we're now seeing coming into trials, which has lost this cohort that potentially would be the best TIL responders, they're not going to get to TIL now because they're responding to CPI, the chances of your overall response rate are lower. Furthermore, post-CPI, there may be new resistance mechanisms in play. So a number of the other checkpoints you've heard about, regulatory T cells, etc. So overall, you would expect your TIL response to be getting worse and worse because of changes in patient profile. And the question is, how do we maximize this group? The patients who we know have NAR-T, neoantigen-reactive T cells in there, who now perhaps are a larger fraction of the overall population that we're going to have to treat with a cellular therapy, how do we optimize the salvageable responders? Do we do it through improved TIL product alone, or can we tackle the tumor microenvironment? So a step back to the work we were doing in Jim's lab, where back in 2004, it wasn't entirely clear how CTLA-4 antibodies were working. We knew they were blocking the inhibitory pathway in effector cells, but there was a great deal of debate about whether they were impacting regulatory T cells. We addressed this question by taking human transgenic mice, so mice that express human CTLA-4, which has a huge degree of sequence homology with the wild-type murine CTLA-4, particularly in the intel domain, but has a number of differences on the surface, which you can differentiate with species-specific monoclonal antibodies. Now, because you can do that, you can reconstitute rag mice either with fully wild-type T-cell compartments, fully human T-cell compartments, or compartments that are one or the other in terms of the regulatory or the effectors, which then allows you to apply species-specific antibodies and look at the impact of the antibody against the effector compartment, the regulatory compartment, neither or both. And that's what we did here. And what we see in green is that if we only target so the blue is the targeting neither. If we only targeted the Treg compartment, we didn't really see much impact at all in terms of the response of the mice and tumor growth. If we targeted only the effector compartment, we did see something. There was a slowing in tumor growth, as you can see here. The mice survived longer. but it was only when you targeted both compartments that you got maximal activity. So CTLA-4 in this murine model was targeting the Treg compartment. And we spent a number of years, Tyler Simpson was a graduate student in Jim's lab, who then continued to work with us after surgery, and I were working at UCL, to demonstrate what was happening. And what was happening was that in the tumor, we were seeing a lack of regulatory T cells when we were treating with our routine anti-CTLA-4 from days 3 to 10. But even if we just gave a single dose of CTLA-4 on the night before we did the harvest of the organs, we saw significant disappearance of the Tregs. So very rapid activity of CTLA-4 in what we subsequently showed was depletion of Tregs within the tumor, as shown here. We then did further work to show that intratumoral Treg depletion was dependent on ADCC and dependent on a specific Fc gamma receptor. So here's your gamma knockout mouse. Treated with anti-CTLA-4, you don't get depletion of the Treg compartment, shown here on a number of mice. And then when we dissected the specific receptor that was involved, it was Fc gamma receptor 4 that was driving the majority of this response. So it was interesting, we'd come up with a new model whereby what was happening in the periphery in the lymph node was that CTLA-4 actually was removing the brakes, allowing T effector proliferation, but also actually Treg proliferation. Something very different was happening in the tumor. And the reason it was different was that Tregs expressed much higher levels of surface CTLA-4 than T effectors, so were better targets for the antibody. And within the tumor, there was this very expanded compartment of CD11b positive, Fc gamma 4 receptor positive macrophages that were actually performing the ADCC. So we had a site-specific activity with depletion of Tregs specifically in the tumor, but getting a large part of the clinical response, or the murine response that I showed you. So that led us to thinking, well, are there other targets that we could prosecute in this way? Particularly CD25. I told you we'd come back to CD25. So if you look at CD25 expression, it's preferentially expressed on tumor-infiltrating FOXP3 cells. So in the blood, it's preferentially on the FOXP3 T cells, and in the tumor, in a number of malignancies, it's preferentially highly expressed in those FOXP3 cells. And if you look at patients receiving PD-1 blockade, remember that's an issue to us, what we don't see is upregulation, we don't see any upregulation of CD25 in the effector compartment, here just looking at CD8, with that treatment. So even with PD-1 blockade, we maintain CD25 on the Tregs, but not on the T effectors. So potentially here we have a target that will differentiate. And a lot of that work then done with an Fc-optimized antibody that would allow depletion of these cells done by Fred Arce in our group at UCL. But we asked further, does the lack of this optimized ADCC fully explain why the CD25 antibodies that we currently had in the clinic weren't actually doing perhaps what we wanted them to? So there were a couple of antibodies, daclizumab and basiliximab. Both of these antibodies were potentially depleters, but also blocked the IL-2 receptor interaction, as did the anti-CD5 clone, PC61, that we used in most murine experiments, where this clone was not effective as a therapeutic. It was very effective as a prophylactic, but not as a therapeutic. And what we wondered whether the Tregs have high levels of CD25 and depleted, T effectors low levels, so they're not depleted, but these are blocking antibodies. So these cells can no longer benefit from the sort of surge of IL-2 that you get. So what would happen if we developed an antibody that was a depleter non-blocker? It would allow potentially IL-2 to access the IL-2 receptor, the high affinity CD25 part. The effectors still have low levels of CD25, so aren't depleted, but now they're fully sensitive to IL-2. So potentially we get the additive impact of IL-2 driving effector proliferation. And there's a very nice cartoon made by the CR UK about the sort of science behind this. So we developed such an antibody for use in our murine experiments. Here's the PC61. Here's our depleter non-IL-2 blocker, and you can see a massive difference in terms of efficacy of that antibody on this particular CD26 murine model. So proof of concept that if we think hard about antibody design, we can resurrect old targets. And here's the crystalline structure, essentially IL-2 binding to the trimeric receptor and the crystalline structure of the antibody, which doesn't bind anywhere near the active site. So finally, I indulged myself somewhat and have gone over my 25 minutes, but I'm still somewhat in time. My concluding remarks. I think neoantigens are interesting. They offer a number of unique opportunities, but also a number of unique challenges as emergent targets for therapies, including the cellular therapies. In my view, they likely account for a large component of clinical response both to checkpoint inhibitors and to TIL, as I've tried to demonstrate today. Now, it's certainly true that most putative neoantigens don't engender corresponding immune response. So it's only a small fraction of predicted neoantigens, less than 10% certainly in virtually all patients, that will engender immune response. It's probably more like 1 to 2%. Patients with essentially lower mutational burden may well have exploitable targets, but the fraction of patients who happen to have that exploitable target will be lower. So commercially, it's less attractive to develop therapies for patients with lower TMBs unless you can develop a pre-screen selection strategy that tells you which patient has the right antigen and inherently the immune response within the tumor. And that is potentially doable. The lack of large numbers of shared or public antigens, even in our experience within the mutational hotspots, does make targeting neoantigens one of the more truly bespoke patient-centric therapies, because essentially the lack of shared antigens means that the development of TCR-based therapies, which at their base work best if you have a public antigen and don't have to make a patient-specific therapeutic each time, is going to be difficult. And finally, given the hostile tumor microenvironment, which probably is more hostile in solid tumors than many of the hematological cancers, and certainly following the positioning of CPI as standard of care in many of those cancers, we will really have to look at additional attempts to impact both cell-intrinsic and cell-extrinsic, the T regulatory population pathways, really beyond the effect we already get with conventional lymphodepletion to actually optimize overall outcomes. Now, one final indulgence for me. We all usually put up slides thanking all the groups we work with, and I certainly would like to thank all the groups I've worked with. One person I never thank is my wife, so I'm going to thank my wife today.